You may sometimes wonder how some of your food, such as tomatoes or strawberries, or some of your materials, such as silk and cotton, are produced on such a mass scale in order to suit the needs of a growing population. Well, I'm here to deliver your answer. Hey there, I'm Lorenz, and today I'll be explaining on how the development of producing transgenic species not only impacts you, but also impacts the field of biotechnology as a whole. Transgenic species are species/organisms that have been genetically modified in order to contain desirable traits or characteristics in order to serve a purpose. Examples of this can be improvement of milk volume in Gujarat cows by taking specific genes from the high-producing Holstein, or the implementation of biological pesticides in a crop in order to not only increase defense against pests, but also combat the rising resistance to traditional pesticide. First of all, however, let me explain how a transgenic species is even made in the first place. The method of producing a transgenic species can be boiled down to three main components: cutting, copying, and pasting. Let's go through it. Firstly, the gene from the source organism that contains the desired trait must be identified. Once this gene is identified, the first step begins: cutting. The gene must be isolated from the rest of the source organism's DNA strand, so enzymes known as restriction enzymes are to be used. The main advantage of restriction enzymes is that they don't cut randomly, rather they precisely cut at different genetic sequences, meaning they are extremely useful in isolating desirable genes from DNA strands. After the gene is successfully isolated or otherwise cut from the source organism's DNA strand, the DNA fragment is then copied by conjoining the DNA fragment to an already cut bacterial DNA plasmid via the use of DNA ligase, a type of enzyme, creating a whole plasmid containing the gene. This hybrid plasmid is then injected back into the bacteria, where the gene-carrying plasmid replicates itself via DNA replication, thus creating more copies of the gene. This process is called gene cloning. This increases the chances of a successful transmission of genes out of sheer numbers. There are various methods of pasting the chosen genes, depending on the purpose of the transgenic species being developed. In the first method, vector injection, involves the use of a vector or medium in order to transfer the gene into the cells of an organism. The most common vector used is a virus, as they are capable of directly injecting or pasting DNA material into the target organism cells. By modifying the virus so they inject a new gene instead of harming the organism, a transgenic organism is able to be produced. This method is usually used in crops or plants. The second method, egg injection, involves directly injecting or pasting the egg gamete of the target species with the new DNA material. The DNA material is pasted into the nucleus of the egg cell, allowing the offspring of the organism to inherit the desired traits. This method is usually used in animals such as goats or cows, as it allows for the organism to successfully reproduce with the gene. So overall, by genetically modifying species by selectively giving them desirable traits such as resistance to disease/pests or increased fat production, mass production of materials and food is possible. One example of a transgenic species being modified for the purpose of mass or more efficient production is the Bt cotton species. Bt cotton differs from the other cotton plants as it has the ability to produce a protein that is toxic to its main pest, the caterpillar of the Helicobacter moth. The protein is non-toxic to humans, as the protein only activates in the digestive system of the caterpillar. The Bt gene that allows for this comes from the bacterium Bacillus thuringiensis, while the method of Bt cotton production follows the vector injection method, as the vector is used to paste the gene is the virus known as Agrobacterium. The species Bt cotton was developed in order to combat rising resistance to pesticides by pests and has proved effective in achieving its purpose. Another example of a transgenic species being produced for the sake of mass production is transgenic goats, modified to produce the protein that creates spider silk. As harvesting and producing spider silk traditionally is not economically viable, the gene that codes for the production of the proteins essential in making this spider silk is pasted into the egg cell of a goat, in which the offspring now has the ability to produce the protein. An example of the egg injection method, as we talked about before. The ability to genetically modify species to suit our needs has, relative to the field of biotechnology, huge implications. The development of transgenic species has arguably benefited society in a huge number of ways. However, there are various ethical issues with the development of transgenic species. One issue is the unknown effects this may have on humans, harmful or not. There are various debates on whether or not GMOs are actually beneficial in the long term, and that they may cause unwarranted side effects to humans. However, these unwarranted side effects are yet to be seen in a widespread manner, so one may say it's unreasonably safe to develop a transgenic species. However, one must remember that with new developments of a field or science, many unforeseen problems may arise. Another ethical issue is the violation to animal rights, as the development of transgenic species is purely for humanity's benefit at the expense of the animals being modified. Transgenic animals may also obtain genetic problems as a result of our tampering, resulting in a permanent alteration of the genetic makeup for the worst. However, one could say that animal rights are being violated every day, such as unsanitary living conditions, systematic harvesting, etc. However, one could also say that despite the constant violation to animal rights, we should not be adding to this list of violations. Despite the many ethical issues and controversies over the development of transgenic species, one cannot deny the many benefits it has brought to society. From increasing resistance against pests, to mass-producing previously non-economically viable products, the development of transgenic species will no doubt continue as long as society benefits from it. And yeah, it's good.